Hey dear students, welcome to the chapter Sexual Reproduction in Flowering Plant. I know that this is somehow board chapter, but we will make sure that you will take interest. See, it is an important chapter because in your board examination, NCRT only you have to focus. Clear? NCRT as well as. You must remember that you are learning for board as well as you are learning for competitive exam. This must you have to remember while learning. And one more thing, make sure that while learning this whole chapter, you have to make notes. Clear? So open your notebook and as well as make notes while learning. We will start sexual reproduction in flowering plant. Let's deal about that. Now in this chapter, we will learn about that what? We will learn flowering structure, that will is development of male and female gametophyte, pollination, types, agencies, pollen pistil interaction, double fertilization, post fertilization, development of seed, apomixis, and significant dispersal, and all about that we will learn in this chapter. So this becomes an important chapter. Let's learn about that. So first we will deal that all flowering plants angiosperm shows sexual reproduction. Flowering plants also known as angiosperm. Clear? So all flowering plant angiosperms show sexual reproduction. Flowers are the sites of sexual reproduction. Clear? So pre-fertilization structures and events we will deal. Now some several hormones and structural changes are there that result in the differentiation or de-differentiation in the plants or floral structure. Clear? So that flower can develop, flower can grow out itself. Clear? Inflorescence is the word. It bears the floral bud and then the flowers. First of all, it happen. First, buds will happen. Then, at last, flower will form. Clear? Now, androecium, also known as a stamen. Clear? It consists of whorl of stamens representing the male reproductive part. So, what is stamen? Stamen is the male reproductive part, or androecium is the male reproductive part. Their number and length are variable in different species. Clear? So, it is not saying that only two in number, three in number, four in number. It may be five in number, it may be six in number, depend on the structure of flower or depend on the species of the flower. Clear? So, a stamen has two parts. Which one is filament and anther? You must know what is filament? Length and slender staff. It's proximal and is attached to thalamus or petal of the flower. Clear? Anther, terminal and typically bilobed. Anther is somehow bilobed, something like that. Clear? This is the structure of flower, something like that. And this is the anther. This is bilobed, and the long slender like that is known as filament. So anther is typically terminal bilobed structure, but each lobe has two theca. That is something like that. That is diathecus. Anther terminal and anthos terminal and typically bilobed. This one is, and somewhat was it is two theca. Clear? Each lobe has two theca. Often a longitudinal groove runs lengthwise separating the theca. Clear? So this is filament and anther. Next we will deal transverse section of anther. The anther is tetragonal structure consisting of four located at the corners. Each lobe consists of two microsporangia. Hence, the diathecous anthers are tetrasporangiate. The microsporangia develops to pollen sacs. They extend longitudinally all through the length of an anther and are packed with pollen grains. Clear? So this is the transverse section of anther. We will deal about what is transverse section. Something like that, a structure of microsporangium. This is the anther. This is the anther. This is bilobed. See, there are two lobes are present. This is bilobed. Clear? So now see the transverse section. This is something like this is pollen grains present in the pollen sacs. Clear? And this is the line of dehiscence. And this is the filament or the stalk. That is cylindrical in part, as we had learned previously. So structure of microsporangium. A typical microsporangium is near circular in outline. Clear? This is circular. It is surrounded by four walls: the epidermis, endothecium, middle layers, and tapetum. The outer three layers give protection and help in dehiscence of anther to release the pollen. The tapetum, this is tapetum, innermost layer, which nourishes the developing pollen grain. This is an important question. becomes and we have competitive exam as Neet or any professional board examination, which nourishes the pollen grain. So tapetum nourishes the pollen grain. Cells of the tapetum contains dense cytoplasm and generally have more than one nucleus. What is tapetum? Cells of tapetum contains dense cytoplasm and generally have more than one nucleus. Clear? So, when the anther is young, a group of compactly arranged homogeneous cell, a sporangious tissue, occupies the center of each microsporangium. Now, see, this is anther which contains pollen sacs, and after that, in inner side, pollen grains are present. Clear? So, this is somehow like that. Now, next, we will deal with microsporangium. See the structure? This is something like that. This is four-lobed, clear? This is connective tissue, epidermis, endothelium, tapetum, which nourishes the pollen grains, the sporogenous tissue, and middle layer, clear? And after intersection, they see epidermis, endothelium, middle layer, tapetum, microspore, and mother cell. Clear? These are pollen grains which is present in the microsporogenesis. Now, as anther develop, as anther develops, each cell of microsporogenous tissue undergo meiotic division to form microspore tetrad. Microspores are arranged in a cluster of four cells. We had seen each one is potential pollen microspore mother cell. Each one has potential to form a microspore mother cell. The formation of microspore from a pollen mother cell through meiosis is called microsporangium. Remember, this is from meiosis, not mitosis. Clear? In the 11th, you had learned what is meiosis and mitosis. So, you must have the difference. So, the formation of microspores from a pollen mother cell through meiosis called microsporogenesis. As the anther matures and dehydrate, the microspore dissociate from each other and develop into pollen grains. Clear? So each microsporangium contains thousands of pollen grains. Clear? Not one or two or three. It consists of thousands of pollen grains. So they are released with the dehiscence of anther. Clear? Next, pollen grain, that is male gametophyte. Now this is a very important question. What is pollen grain? See, generally spherical size 20 to 50 μm in diameter. Clear? 20 to 50 μm in diameter. Cytoplasm is surrounded by a plasma membrane. Pollen grain cell wall is made up of two types of layer. One is exine, that is outermost, and one is entine. Clear? You must remember exine and entine. Important question comes in the Neet. So the outer layer, that is hard layer exine, made up of sporolanin. Must remember sporolanin is one of a pollen grain that it has a high resistant organic material. It can withstand with high temperature and strong acids and alkali. Clear? So this is exine. One more thing, enzymes cannot degrade the sporolanin. Clear? So enzyme is a apertures called germ pores, where sporolanin is absent, but pollen grains are preserved as fossils due to presence of sporolanin. Clear? Why? Because it has a high resistant organic material. That's why it can be preserved for the fossils after a long year by year. Clear? So exine exhibit patterns and designs. Clear? So now this is exine. What is entine? The inner wall is entine. It is a thin continuous layer made up of cellulose and pectin. Very important. Entine is made up of cellulose and pectin. But exine is a hard layer, and exine hard outermost layer, but it is made up of sporolanin, which is high resistant organic material. You must have to remember, make notes with me. Clear? So this is all about pollen grain. Next, what is mature pollen grain? It contains mature pollen grains contains of two cells. Which one? Vegetative cell and generative cell. Vegetative cell, it's bigger, has abundant food reserve and large irregularly shaped nucleus. Clear? But generative cell is what? It is small, floats in the cytoplasm of the vegetative cells. Means generative cells is small but floats in the vegetative cells of cytoplasm. It is spindle in shape with dense cytoplasm and a nucleus. It of angiosperms, pollen grains are shed at the two-celled stage. In others, the generative cells divide mitotically, not meiotically, mitotically to give rise to two male gametophytes. Like that, before pollen grains are shed, three-celled stage. We'll deal what is three-cell, two-cell, but just learn Generative cells divided mitotically. Clear? The shed pollen grains have to land on the stigma before they lose viability. Viability is known as consistency, their work potential. This is known as viability. The viability period of pollen grains depends on the prevailing temperature and humidity. Depend on the temperature and humidity. So, pollen viability of pollen grains of some cereals, rice, wheat, etc. is 30 minutes. What? Only 30 minutes. Clear? You must have to remember the time. So viability of pollen grains of some cereals like rice, wheat is 30 minutes within their release. Clear? Some members of like Leguminosae, Rosaceae and Solanaceae have viability for month, 1 month or 2 months, somehow depend on the potential. Clear? So this is important. Viability of pollen grains is 30 minutes and mitotically divide into two male gametes. Clear? Now see the structure. This is pollen grain, this is vacuoles, and this is nucleus. This is asymmetrical, the spindle formed, and after that it forms into different stages, that is vegetative cell and generative cells. Clear? Now, what is the economic importance of pollen grain? Why it is necessary? You know, anyone? See, they are rich in nutrients. Pollen tablets are used as a food supplement. You have heard that pollen tablets are used for food supplement. If someone is weak body or weak in feminine, that is feminine or male muscular body tone, so they can use pollen tablet, but as a prescription of doctor. So pollen tablets and syrups increase performance of athletes and racehorses. Clear? Pollen grains can be stored for years or years in what? Liquid nitrogen. But what's the temperature? Minus 196 degrees Celsius. Very important. They are used as pollen banks, similar to seed banks in crop breeding programs. Pollen grains of some plants, example, carrot grass. You have seen carrot grass? You must have Parthenium carrot grass. Are allergic for some people. Clear? So it leads to chronic respiratory disease like asthma, bronchitis. Clear what I'm telling? So pollen tablets and syrups are used for the high or increased performance of athletes and racehorses. So pollen tablets can be used as a supplements to increase metabolism, to increase density of human individuals. Clear? So also they can be used as a pollen banks similar to seed banks, like that only in crop breeding programs. Clear? One more thing, what we had learned, pollen tablets or pollen grains can be preserved in what? Minus 196 degree Celsius or temperature में. Clear? Minus 196 degree Celsius में it can be preserved. Clear? So you have to learn. This is very important. See, I had made very clarified notes from the NCERT and from the different reference source also. So make sure you must have to make all the points, because all the points are very important. It is not necessary that it will be of extra. No, it is very important. All the notes and all the points I had taken from the NCERT and as well as reference from the another books of biology. Clear? So you must have to know, make notes. Now, gynoecium, that is pistil. It represents the female reproductive part of the flower. It may consist of single pistil, that is monocarpellary. Gynoecium is female reproductive part and the testamen is male reproductive part. So gynoecium pistils represent the female reproductive part of flower. It may consist of single pistil, that is monocarpellary, or more than one pistil, that is multicarpellary. In multicarpellary, the pistil may be fused together, that is syncarpus. Very important. Now you will say what is syncarpus, what is apocarpus. This is very important. See, gynoecium is female reproductive part. If single pistil, called monocarpellary. If more than one pistil, that multicarpellary. Now see the difference, single pistil called monocarpellary, more than one pistil, or double pistil called multicarpellary. Same like that, in multicarpellary, pistils are fused together, that is syncarpus. Clear? If they are fused, called syncarpus. But if they are free, called apocarpus. Clear? So hibiscus pistil, multicarpellary syncarpus pistil is of papaver. This is important example. And multicapillary apocarpus gynoecium of Michaelia. Very important. You must remember. You must remember what is encarpus, what is apocarpus, what is multicapillary, what is monocapillary, and what is multicapillary. Clear? So this is the important. This some terms you have to learn more and more after sometimes or after the next chapter. So you must remember the meaning of the terms. What is multicapillary? What is encarpus? What is apocarpus? Clear? Now each pistil has three parts. Now how three parts? Stigma, style, ovary. See, this is ovary, this is thalamus, this is style, and this is stigma. Clear? Now see the stigma portion, see the carpus, something like that. See the structure of ovule. Now this is in the ovule. Huh. One more things in the ovulation, in the ovule of this pistil, you have to make diagram. Very important. It will come to the board examination. This is hilum, starting point funicle, micropyle, micropylar pole, outer integument, inner integument, nucleus or embryo sac or chalaza pole. Clear? So this is about ovulation. Now, the stigma is what? Stigma is what? Now see the structure of a stigma. It is landing platform for pollen grains. It is an elongated, slender part beneath the stigma. Ovary, it is the basal bulge part of the pistil. Inside the ovary is the ovarian cavity. In the inside of ovary, it has a present ovarian cavity that is known as locule, in which the placenta is located. Clear? Arising from the placenta are the ovules, that is megasporangium. The number of ovules in an ovary may be one. Wheat, paddy, mango, etc. They can be. So, to many, that is pepper, watermelon, orchids, etc. These are what? Number of ovules it can be present for the different seeds or different fruit structure. Yeah, so this is the parts of the pistil. What? Stigma, style, and ovary. Clear? You must have to make notes and must have to make diagram. Very important in your board examination. In each and every question, if you thought that diagram is necessary, then you must have to draw diagram for get to get full marks. Yeah. So now, microsporangium, that is ovule, it is a small structure attached to the placenta by means of stalk called funicle. The junction where the body of ovule and funicle fuse. It is known as hilum. Each ovule has one or two protective envelopes called integuments, that is outer integuments and inner integuments. We had seen in the structure. So, integuments encircle the ovule except the tip where a small opening micropyle is present. That is micropyle, not P. Opposite of the micropylar end is called chalazial or basal part. Enclosed with the integuments, there is mass of cells called nucellus. Its cells contains reserved food materials. The embryo-sac female gametophyte is found within the nucellus. An ovule generally has a single embryo sac found with megaspore to meiosis. Now, what see difference between meiosis and meiosis? In the meiosis, four daughter cells are formed. But in mitosis, two uh, daughter cells are formed. See, learn the difference. Two daughter cells are formed. Clear? So this is the difference. Now, see the megasporangitis. That is micropylar and nucellus, megaspore mother cell. Clear? Micropylar and nucellus, megaspore diet. Here, this is the megaspore tetrad. See, first two cells are formed. That is two microspore diet. But when three form, megaspore tetrad. Clear? Yes, it becomes four, not four chambers, so it calls tetrad, not triad. Clear? So now see, these are formation of embryo, something like that. Megasporangial formation. Fourth daughter cell becomes two daughter cells, then four. Now see, this is the synergids, this is X cells, central cells, polar nuclei, and this is known as antipodal cells. Clear? Now see chalazial ants, this is antipodals, polar nuclei, central cells, egg, synergid, filiform, apparatus, this is micropylar ant. All the structures I had taken from the NCERT only. Clear? So you must remember why NCERT structure is important in each and every questions becomes for competitive exam or Neet or any professional board exam only becomes from the NCERT only. Clear? Now see, what is megaspore mother cell? It is the formation of megaspores from the megaspore mother cell. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. It is large cell with dense cytoplasm and a prominent nucellus. The MMC undergo meiotic division. MMC is what? Megaspore mother cell undergo meiotic division means two daughter cells are formed. It results in the production of four megaspore. Clear? So this is something like that. Female gametophyte embryo sac. In the majority of flowering plants, one of the megaspores is functional while other three degenerate. The functional megaspore develops into female gametophyte, and this method of embryo sac formation from a single megaspore is known as monosporic development. Now what happens? When the single megaspore formation is formed from the embryo sac, that is known as monosporic development. Clear? So the nucleus of functional megaspore divides mitotically to form two nuclei. Clear? To form two nuclei. Then they move to the opposite poles, form two nucleate embryo sac. Now the nuclei again divides two times. Then it becomes double. So it forms four nucleate and later eight celled stages. Here now seven celled, eight nucleated, seven celled and eight nucleated. Very important repeated question in the NEET examination. Clear? So this is of seven celled or eight nucleated. Clear? So this is how female gametophyte embryo sac is formed. Now this division are strictly free nuclear. That is, nuclear divisions are not followed immediately by the cell wall formation. After the 8 nucleate cell, the cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. Now, the 6 of the 8 nuclei are surrounded by cell walls and organized into cells. Remaining 2 nuclei, that is polar nuclei, are situated below the egg apparatus in the large central wall. Now, which is important, seven celled and 8 nucleated form after at last a formation from the embryo sac. This is important only. Now, distribution of cells within the embryo sac. See, a typical mature embryo sac is divided and it is mature to form seven celled, 8 nucleated. Clear? Very important, very, very important. Always a repeated question. Three cells are grouped at the micropylar end and form egg apparatus. It consists of two synergids and one egg walls. What? It consists of two synergids and one egg cell. Clear? Synergids have special cellular thickness at the micropylar tip to form filiform apparatus. What? It helps to guide pollen tubes to the synergid. Clear? So, synergids have a special thickness or a special cellular thickness at the micropylar tip to form filiform apparatus. And what they will do? Filiform apparatus guide pollen tubes into the synergid. Clear? So, three cells of the chalazal ends known as antipodals, and the largest central wall has two polar nuclei. Clear? Now see, what is pollination? Pollination is what? It is the transfer of pollen grains from the anther to the stigma of a pistil. Some external agents helps the plants for pollination. External agents such as bees, vines, ears, these are the external agents. Now, depending upon the source of pollination, pollination is of three types. Which one? Autogamy, xenogamy, and geitonogamy. Autogamy is what? Self-pollination. Geitonogamy is cross-pollination. Xenogamy is what? Cross-pollination. And geitonogamy is for same plant but another flower. Okay. So this is somehow what pollen grains. Pollens are pollinators. Pollinators present like bee, and it grows to the stigma. This is pollination occur. Clear? Now see. What is autogamy? Autogamy is self-pollination. In this, pollen grains transferred from the anther to the stigma of the same flower. In flowers with exposed anther and stigma, complete autogamy is rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity. Also, anthers and stigma should lie close to each other. Plants like viola, common palsy, oxalis, and camelina produce two types of flowers. like chesmogamous flower and clistogamous flower. Very important. These two types of flower, chesmogamous flower, they are similar to flowers of other species with exposed anther and stigma. Clear? So they can expose anther and stigma, but in the clistogamous flower, they do not open at all. They cannot expose their anther and stigma. What I'm saying? Clistogamous flowers cannot open. They do not open at all their anthers and stigma. But chesmogamous flowers are openly. They can expose their anthers and stigma. So anthers and stigma lie close to each other. They are autogamous. When anther dehisces in the flower pollen grains, buds come in contact with stigma for pollination. Now, clistogamous flowers produce assured seed set even in absence of pollinators. Clear? So, this is all about autogamy. Now, we will read xenogamy. Xenogamy is what? Cross pollination. See, this is chesmogamous flower. This is clistogamous flower. This is not open at all, and this can open at all, that is anthers and stigma. Now, xerogamy in this pollen grains transfer from anther to the stigma of a different plant. This is cross pollination. It brings genetically different pollen grains to the stigma. Clear? Now, geitonogamy is what? In the pollen grains transfer from the anther to the stigma of a pollen of anther flower of the same plant. It is functionally cross pollination involving a pollinating agent, but it is genetically similar to autogamy since the pollen grains come from the same plant. What I'm saying? Geitonogamy is what? Pollen grains transfer from anther to a stigma of another flower of same plant. Another flower of same plant, that is geitonogamy. Functionally, it is cross-pollination involving a pollinating agent like bees, air, wind, etc. But it is genetically similar to autogamy since the pollen grains comes from the same plant. Why it is autogamy? Because it has a pollen grains comes from the same plant. Clear? So this is the geitonogamy. Yes, now see agents of pollination. Do you know what is pollination, how and what are the agents are present? So we'll deal about that agents of pollination. So abiotic agents, that is presence of wind and water. This is the main two agents are present for the respect of pollination. So pollinated by wind, that is the pollination of wind or the pollination by wind is known as anemophily. Clear? More common in abiotic agents. Wind pollinated flowers often a single ovule in each ovary and numerous flowers packed into an inflorescence. For example, corn cob. The tassels are the stigma and the styles which wave in the wind to trap pollen grains. Clear? So this is corn cob. Search in the Google corn cob, and so that and check tassels are the stigma and the style wave which in the vine to trap pollen grains. So wind pollination is quite common in grasses. So in grasses, what will happen? Wind pollination occur. See, effective pollination ways. See, this is like something like that. Pollen grains occurs, fall down. The flowers produce enormous amount of pollen. The pollen grains are light, non-sticky, so that it can be transferred here to there or one place to another place by wind currents. They often possess well-exposed stamens for easy disposal of pollen into wind currents. Large feathery stigma to trap airborne pollinators. Clear? So they have large feathery stigma. Why? To trap airborne pollen grains. Clear? So this is about ways for effective pollination. What will occur? First, it is light, non-sticky, so that it can move here to there. Or it is well exposed stamens. Why? For easy dispersion of pollens in the ground. Clear? Into the vine current. So these are the effective pollination occurs. Now see pollination by water, that is hydrophily. See, this is female flower and this is stigma. This is male flower and this pollination occur. Pollination of water called known as hydrophily, but pollination as white is known as anemophily. Clear? So it is quite rare. It is limited to about 30 genera. Most monocotyledonous, not dicotyledonous. For example, Welsneria, Hydrilla, freshwater, or Zoostera, that is marine seagrasses, etc. This is very important example. You must have to learn pollinated by water, that is hydrophily. Clear? So as against this, water is a regular mode of transport for the male gametes among the lower plants. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need for water for the transfer of male gametes and the fertilization. In the female flower, reach the surface of water by the long stalk, and male flowers of pollen grains are released on the surface of water. So what will happen? They carried by water currents and reached through the female flowers. In sea grasses, what will happen? Female flowers remain submerged in water so that pollen grains are long and ribbon-like. So they carried inside the water and reached into the stigma. The pollen grains of most of the water pollinated species have a mucilaginous covering, some sticky or some smoothing like that, mucilaginous covering to protect from the wetting. Clear? So this is all about pollination by water and wind. Now, not all aquatic plants are hydrophilic, cannot use hydrophilic, but most of aquatic plants such as white water hyacinth, water hyacinth, what is terror of Bengal, water lily, they are hydrophilic. The flowers emerge above the level of water for entomophily or anemophily. Wet and water pollinated flowers are not very colorful and do not produce nectar. Clear? So this is about wind and water pollinated flowers. Now we will learn biotic agents. Majority of flowering plants use animals as pollinating agents. Clear? For example, bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds, hummingbirds, bats, primates, that is lemurs, arboreal, tree dwelling, rodents, reptiles on the like zecco lizard or garden lizard, etc. Pollination by insect, that is pollination by insect also known as what? Entomophily. Clear? Pollination by air, pollination by wind, pollination by water, pollination by insect. Pollination by insect is known as what? Entomophily. Remember, particularly bees is more common. Clear? So often flowers of animal pollinated plants are specifically adopted for a special particular species of animal. Clear? So these are biotic agents by somehow insect or something other organism like that. Now, features of insect, that is pollinated flowers. What are the features? Large, colorful, fragrant, and rich in nectar. Nectar and pollen grains are floral rewards for pollination. When the flowers are small, they form inflorescence to make them visible. The flowers pollinated by flies and beetles secrete foul odors to attract these animals. The pollen grains are generally sticky. When the animals comes in the contact with the anther and the stigma, its body gets pollen grains. When it comes in contact with the stigma, it results in the pollination. Some plants provide safe places, floral reward to lay eggs. For example, Amorphophallus. Remember, tallest flower of 6 feet. So some are very tallest flower. A moth species and the plant yucca cannot complete their life cycle without each other. So the moth deposits its egg in the locule of ovary. The flower gets pollinated by moth. The larvae comes out of the egg as a start developing seeds. Many insects consume pollen or nectar without bringing about pollination. So they are called pollen or nectar rovers. Clear? So this is all about the features of insect. This is not so much important, but you have to remember tallest flower of 6 feet, that is Amorphophallus. Otherwise, you just remember the features of insect. Now, it is important what is outbreeding devices. What is outbreeding devices? Now, see, hermaphrodite flowers can undergo self-pollination, continued self-pollination, resulting in inbreeding depression, loss of flowers. So, if continuation of self-pollination occur, what will happen? Inbreeding depression. To avoid self-pollination and encourage cross-pollination, there are some devices. Or we can say to the question, what the question become, what will you use to avoid inbreeding depression? So in the inbreeding depression, what you will do? You will do cross pollination to reduce inbreeding depression. Clear? What's the question? What you will use to avoid inbreeding depression? You will use cross pollination. Clear? So first, avoiding synchronization. Matlab, here
The pollen is released before the stigma becomes receptive, or stigma becomes receptive before the release of pollens. It prevents autogamy. Arrangement of anther and stigma in different position also prevent autogamy. Self-incompatibility. It is a genetic mechanism to prevent self-pollination from the same flower or another flower of the same plant from fertilization by inhibiting pollen germination or pollen tube growth in the pistil. Production of unisexual flower. If male and female flowers are present on the same plant, that is monoecious, for example, castor and maize, it prevents autogamy but not heterogamy. This is all very important. Very important. It prevents autogamy but not heterogamy. In dioecious plant, for example, papaya, male and female flowers are present on different plants that is dioecious. This prevents both autogamy and heterogamy. Here, autogamy, heterogamy, this flowers you must have to learn. Note it down, very important. Now, this is pollen pistil interaction. See, this is all plasma membranes, allergic, filiform apparatus, something like that. Male gametes, vegetative nucleus. This is male gametes. And this is all about pollen pistil. See, pollen tube, antibody cells, pollen nuclei, excel and synergy. Now, what is it? Is a process in which pistil recognize compatible or incompatible pollen through the chemical components produced by them. If the pollen is the right type, the pistil accept it. But if not right type, it cannot accept it, it will reject. And if pollen is correct, it promotes post-pollination events. Pollen grains germinate on the stigma to produce the pollen tube through one of the germ pores. The content of pollen grains move into the pollen tube. Pollen tube grows through the tissue of a stigma and a style and reaches the ovary. If the pollen is incompatible, called wrong type, it rejects. Pistil rejects pollen by preventing pollen germination on a stigma or pollen growth in the stem. Clear? Means if it is right type, then only it will accept, or if it is wrong type, it will reject. Clear? So a line continuous dialogue between the pollen grain and the pistil result in pistil ability to recognize the pollen to accept or reject it. Clear? This is a method, this is a right type and wrong type. You must have to remember about the pollen pistil interaction. Clear? Same as like that, if two individuals, male and female, interact with them, if male or female is on that type, then also then only they can introduce themselves. For example, you have one friend and you have another friend. If she looks like your type, then only you can talk. Or if you will think that if you will think that we have a good friendship or we can different understanding, then we will not meet. But if you think that we have same understanding, same type, so we can be friends. Clear? Something like that only, right type and wrong type of pollen pistil interaction. Now, in some plants, pollen grains are two-shaded, that is two-cell condition, that is vegetative and generative. Already I saw. In such plant, generative cells divide and form two male gametes during the growth of pollen tube. But in three-cell condition, pollen tube, remember, in three-cell condition, pollen carry two male gametes in the beginning. So pollen tube reaches the ovary, that enters the ovule through micropyle and then enters one of the synergid through the filiform apparatus. Now, filiform apparatus present in the micropylar part of the synergid guides the entry of pollen tube. A pollen breeder can manipulate pollen pistil interaction. Here, even an incompatible pollen to get desired hybrid. So, only the plant breeder can accept all these things. It can manipulate, it can interact, it can do all things. Clear? So, a pollen breeder can manipulate pollen pistil interaction, even an incompatible pollination to get desired hybrid. Clear? So, this is all about that. Now, artificial hybridization. What is artificial hybridization? You will see in the crop improvement. It is a crop improvement program in which desired pollen grains are used for pollination. This can be achieved by different techniques. For example, see, for example, removal of anthers from the bisexual, removal of anther from the bisexual flower buds in female parent before the anther dehiscence using a pair of forceps. Emasculation in the meaning of removal of anthers. Emasculation meaning what it means? Removal of anther. But what do you mean by bagging? Here, emasculated flowers are covered with a suitable bag made up of butter paper to prevent contamination of the stigma with unwanted pollens. Somehow, this is a flower. This is like that, and you had done. This is butter paper. You just cover it out and to prevent contamination. When the stigma does receptivity, mature pollen grains collected from anthers of the male parent and dusted on the stigma. Then the flowers are rebagged and allowed to develop fruits. Clear? So for unisexual flower, there is no need for emasculation or bagging. Female flowers, buds are bagged before the flowers open. When the stigma before receptive becomes receptive, pollination is carried out using the desired pollen and the flower rebag. Clear? So this is all about the emasculation and bagging, the technique used in the artificial hybridization. You have to remember. Now what is double fertilization? See, after entering one of the synergids, the pollen tube releases two male gametes. Clear? Where in the cytoplasm of synergid. Now, one male gamete move towards the egg cells and fuse its nucleus to form zygote. Clear? See, double fertilization, what will happen? See, when it enters to the synergid, what will happen? Pollen tube releases two male gametes. This is one male gamete and this is another male gamete. Check. Now, one male gamete moves towards egg cell. One moves towards egg cell and fuse with the nucleus. So egg cell fused with nucleus, what it will form? It will form zygote. And what is zygote? Zygote is a diploid cell. Now, another male gamete moves toward the two polar nuclei. Where it will go? Two polar nuclei. Where it is located? Located in the central cell and fuses with to form triploid primary endosperm nucleus. That is spin. Clear? It fuses with what? Central cell. And what it will form? Pen. That is triploid primary endonucleus. Clear? This is the so as it involves fusion of three haploid nuclei. This is known as triple fusion. Very important. Since two types of fusion, syngamy and triple fusion, takes place in embryo sac. Now in the single embryo sac, two types of fusions are there. One is egg cell with nucleus form zygotes, and another one is two polar nuclei nucleus with central cell fused to form pen. That is triploid primary endosperm nucleus. So, as cells involvement of two types of fusion, that is syngamy and triple fusion. Syngamy is what? This is one type syngamy, this is triple fusion. In a single embryo sac, known as double fertilization. Clear? As its events unique to flowering plants. Clear? Very important. Double fertilization. Remember? Now, this is something like that. Degenerating cells, zygote. This is primary endosperm cells. And after fusing primary endosperm nucleus, that is pen. Degenerating antipodal cells, this is from zygote, globular embryo, heart-shaped embryo. This is suspensor, plumule, this become mature, cotyledon radical. This whole important, very important. Clear? So, the central cell after triple fusion become primary endosperm cell. PEN becomes, after triple fusion, what will become? Primary endosperm cell. That is what? After triple fusion. Clear? And develops into the endosperm while the zygote develops into an embryo. Clear? So this is all about that. Very important, very, very important. Comes to the board as well as your needs summary. Now, post-fertilization structures and event. See, post-fertilization endosperm and embryo development, maturation of ovule into seeds and ovary into fruit. Now, endosperm development. The primary endosperm cells divides repeatedly and form a triploid endosperm tissue. Now, endosperm cells are filled with reserved food material. They are used for developing embryo. Now, see, in the most of the common type of embryo development, the pen undergoes successive nuclear division to give rise to free nuclei. Now, see, this stage is known as prenuclear endosperm. The number of free nuclei vary greatly. It can be different in number. So, the formation of cell wall causes the endosperm to become cellular. Now, in coconut, the surrounding white kernel is cellular endosperm, while the soft coconut water is free nuclear endosperm, made up of more than 1,000 of nuclei. Very important. Surrounding outermost layer white kernel is cellular endosperm, while soft coconut water, hard coconut water, soft coconut water is free nuclear endosperm made up of more than 1,000 nuclei. Clear? Now embryo development. See, this is plumule, cotyledon, hypocotyl. This structure is very important. You can forget. Just remember the structure. Make practice. Now, embryo develops at the upper end of the embryo sac where the zygote is situated. Most zygote divide only formation of certain amounts of endosperm. This is an adaptation to provide nutrition in the developing embryo. Through the seeds differ greatly, the embryogeny development is similar in monocots and dicots. The zygote give rise to the proembryo and subsequently to the globular heart-shaped and mature embryo we had learned previously. Clear? So this is about embryo development. What will happen? Zygote, first will form zygote, then it will give heart-shaped globular and mature embryo. Clear? So through the seeds differently, it becomes embryogeny. Early development is similar in monocots and dicots. Clear? Now dicotyledonous embryo. It has some scutellum, coleoptile, shoot apex, septiblast, radical root cap, and coleorhiza. Now, dicotyledonous embryo is embryonal axis have two cotyledons. The portion of embryonal axis above the level of cotyledon in the epicotyl, which terminates with the plumule, that is the stem. The cylindrical portion below the level of cotyledons is that terminates with the radical tip. The root tip is covered with a root cap. Clear? So this is dicotyledonous embryo which have two cotyledons. Now, monocotyledonous embryo passes only one cotyledon, something like grass family. The cotyledon called scutellum. It is situated lateral to the embryonal axis. At its lower end, the embryonal axis has radical root cap closed in the coleorhiza. Clear? Previous diagram it is comes into the monocotyledonous embryo, but you see an undifferentiated sheath. Portion of embryonal axis, absence of level of attachment of scutellum is the apicotyl. Now it has shoot, apex, and few primordia enclosed in the coleoptile. Previous structure is comes into the monocotyledonous embryo, and this structure goes to the dicotyledonous embryo. But see, this is the dicotyledonous, something like that. Coleoptile, plumule, radical, coleorhiza, the seed coat, and all are present. Now, seed from ovule. What will form? Seed is a fertilized ovule from inside oops. It is a final product of sexual reproduction. Clear? It consists of seed coat, cotyledonous, and embryo eggers. The cotyledons are simple, generally thick and swollen due to storage food as in legumes. Now, mature seeds are of two types, non-albuminous and albuminous. Very important, very important. Non-albuminous seeds have no residual endosperm. Why? Because it is completely consumed during embryo development. For example, pea, groundnut, beans. Remember, imagine their structure seed. But albuminous seed, it has a retained part of endosperm as it is not completely used in embryo development. For example, wheat, maize, barley, castor, coconut, sunflower. This is all about that. Now, true fruit and false fruit, we will learn what is true fruit and false fruit. Now, see, oil reserve of groundnut is present in cotyledonous. Occasionally, in some seeds like black pepper, wheat, etc., remnants of nucleus are also persistent. It is called perisperm. Integuments of ovules harden as tough protective seed coats. It has a small pore through which Oil and water enter into the seed during germination. As the seed matures, the water content is reduced and seed becomes dry. 10 to 15% moisture by mass. The general meiotic activity of the embryo slows down. You know, so the embryo may enter a state of inactivity. This is known as dormancy. If a favorable conditions are available, activate adequate moisture or oxygen and suitable temperature, then they can germinate. Yeah, otherwise does not, becomes dry. Now, seed from ovary. The ovary develops into a fruit. Transformation of ovules into seeds and ovary into fruit produce simultaneously. But the walls of ovary develops into pericarp, that is wall of fruit, and the fruit may be guava, orange, mango, or dry fruits such as groundnut and mustard. Fruits are of two types, true fruit and false fruit. False fruit, very important. True fruits which develop only from the ovary and other floral plants generate and fall out. But false fruit, then this thalamus also contributes to fruit formation. Clear? So this is known as false fruit, like that apple, strawberry, cashew. These are false fruit. But what is true fruit? Watermelon, lemon, cherry. These are known as true fruit. Clear? Why? Why true fruit? Because only fruit develops from the ovary. But in the false fruit, it develops from the thalamus also. This is the huge difference you must have to remember. Very important. Now, what are the advantages of seeds? Since pollination and fertilization are independent of water, seed formation is more independent. Seeds have better adaptive strategies for dispersal to new habitats and help the species to colonize in the other areas. They have food reserves. So young seedlings are nourished until they are capable of photosynthesis. The hard seed coat protects the young embryo. Being products of sexual reproduction, they generate new genetic combination leading to variations. Dehydration and dormancy of mature seeds are crucial for storage of foods, clear, or seeds. So it can be used as throughout the year and year for to raise crop for the next session. And farmers are doing only like that. Clear? So they can store a year by year for the seeds. Now, what are the viability of seeds after dispersal? This is important. In few species, the seeds lose viability within a few months. Some of many seeds live for several years. Some can live hundred of years. For example, the oldest is that lupine, very important, Lupinus arcticus, extricated from Arctic tundra. Clear? The seed germinated and flowered after an estimated record of 10,000 years of dormancy. Now, 2000 years old viable seed, that is date palm, Phoenix dactylifera. This is discovered during archaeological excavation of King Herod's palace near the Dead Sea. Search in the Google. You can see about the date palm. In some plants, fruit contain very large number of flowers. For example, orchid, orobanche, and striga. This is I taken from the reference. It is important. Fruits are one such category and each fruit contains thousands of tiny seeds. Clear? So these are the fruits of orchids, orobanche, astraga, that is Phoenix, dactylifera, that is date palm. Here, 1,000 to 2,000 of year old viable seed can be placed. That is lupine arctic, that is old, one of the oldest from come from the Arctic tundra. Clear? So this is important viability of seeds after. Now we have the last topic, last but not least, apomixis and polyembryony. Now apomixis is the production of seeds without fertilization. Clear? Some species of grasses. One is missing, that is Astraceae. It is a form of asexual reproduction that mimics sexual reproduction. So species of Astraceae and grasses. Now development of apomictic seeds. In some species, the diploid egg cells is formed without reduction division and develops into embryo without fertilization. In many species, like many citrus and mango varieties, some of the nucellar cells surrounding the embryo sac divide, protrude into the embryo sac and develop into embryos. Now, in such a species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is known as polyembryony. More than one embryo known as polyembryony. Now, importance of apomixis in hybrid seed industry. If the seeds collected from the hybrids are sown, the plants in the progeny will segregate and lose hybrid characters. Production of hybrid seed is costly, hence cost of hybrid cells is also expensive for the farmers. If the hybrids are made into apomix, then there is no segregation of characters in the hybrid progeny. So then the farmers can keep on using hybrid seeds to raise new crop after a year by year. So this is all about the chapter sexual reproduction in flowering plants. Hope you will enjoy, but make sure learn notes for the board's examination. After that, revise properly. Yeah, we'll do next chapter. Wishing you all the best. Good luck.